Switching gears just a little bit. I'm Justin Blethrow. I'm part of the product team at Plasmidsaurus. I've been with the company about, 7 or 8 months now, and it has been a fantastic whirlwind adventure developing what I'm going to tell you about here. Before I was at Plasmidsaurus, I was at PacBio. I was the product manager for a lot of the long-read instrumentation you may have used or have heard about. And long ago, I was at Thermo developing Orbitraps in the proteomics space. So I spent a lot of my career developing sort of heavy iron instrumentation, and now I'm at a sequencing company. And why? Well, the reason I was motivated to join, I came in and talked to Jim and Mark, and they shared some of the vision that Jim went into in the beginning there. And I realized this isn't just any other sequencing company, right? There's a lot of service providers. They all offer fairly equivalent products in a lot of ways. But here was a company that was interested in really transforming the way that people did work and had built the business on a product that I greatly wish that I had in grad school. I remember a lot of mornings doing 200 mini preps and trying to get that done in time for some coffee or at least lunch. And I wouldn't have had to do any of that. And I came in and I asked, well, what would I be working on? And we had notions about what this product would be, but it was still had very broad constraints on what it would be. So I had an opportunity to really shape this. And so the combined vision of the company and then the vision, at least the nascent vision for what we were going to do with RNA-Seq was extremely compelling to me. And just to spend a few more minutes talking about some of the thought process that went into this, we wanted to do something in differential gene expression, right? We didn't want to just have another commodity sequencing product. Like the plasmid service, we wanted to come up with something that was truly enabling and accelerating for customers across as broad a swath of biology as we possibly could. And differential gene expression was the theme we had in mind. Anytime you're making some kind of biological change to cells or tissues, whether that's gene editing or drug treatment or just some kind of condition, you want to know how the biology is responding, right? You want to know what genetic circuits are turning on, what pathways are becoming active or becoming inactive. And you want to understand, ideally, as deeply and broadly as you can, what's going on in the cell. And just to cast back to the slide Jim showed here, this lesson learned that sometimes the things you're not seeing are really important, right? Sometimes backbone mutations in your plasmid might be causing problems that could derail your whole project if you don't know about them. Well, similarly, in gene expression, if you're only taking a very targeted view, for example, through qPCR or maybe phenotypical assays like fluorescence microscopy, you're only getting a narrow view and you may be missing things that are really important. Maybe you're making, just as a random example, maybe you're making a transgenic line and you see through either of these mechanisms or many others you can do, Western blots, that your target gene is present, your transgene is present at a decent level. But you may not realize that, well, it integrated somewhere bad and broke something else that was really important. Or any of a whole host of other possible things that could be going on that you may want to know about. And so to get at that broad picture, traditionally, really you need to do very high bandwidth, very large scale assays like NGS-based RNA sequencing or once upon a time arrays. And those give you the broad view, but they tend to be very expensive and very slow. Legacy service providers often take a month or more, sometimes several months, to get data back from experiments like this. And alternatively, you can do it in-house if you want to go through the capital equipment expenditure and spend a lot of time getting your second PhD in operating this expensive equipment, essentially. So our vision for this product was something that would come in here at the sweet spot in both of these dimensions, that would let you get genome scale insights with the sort of speed and accessibility, and I use accessibility as a proxy for both cost and the amount of work you have to put into the process of something like qPCR, or at least the higher end of qPCR where you're looking at more than one or two genes. And of course, the product also needed to meet our general product design philosophy that Jim explained and I've indicated a little bit, but the product needs to be very simple from the user perspective, while sophisticated in what it actually does. It needs to be very fast, provide high quality, accurate data. We can't compromise on the fundamentals. The results need to be intuitive so that you can get insight out of it right away rather than having to, well, now what do I do? Find a bioinformatics expert or spend a bunch of time learning to be in bioinformatics, or maybe GPT-5 can help me write some scripts. And it needs to be really affordable. So to cut to the chase a little bit, this is what we came up with, and I'll show you the top line specs, and then I'll go into more detail on what we're doing or how it works and what design decisions we made. So this is a genome-wide quantitative gene expression analysis assay that we turn around in about 3 days in North America at least, and it'll be 3 days in the rest of the world pretty soon. Outside North America right now, it's about 4 or 5 days. And the all-in pricing on this is $50 per sample for academic and other nonprofit customers, and $80 per sample for commercial ventures. And people often ask, well, does that include the analysis? Does that include the library prep? Yeah, that's truly all-inclusive. So more specifically, what is it and how does it work? When we originally conceived of this, we thought that the vast majority of market would be in service of cell lines, and then so some of the assumptions were designed to target that, and also to make the service as comprehensive end-to-end as possible. So Jim mentioned how we like to try to take responsibility for as much of the workflow as we can. We see that as being mutually beneficial because it takes burden off of you, and it allows us to automate and standardize as much of it as possible, which reduces the chance of human errors and such. So in the maximal case here, you can submit cultured cells, about 100,000 or 200,000 cells, or plant protoplasts if you have them available, in a product called ZymoShield. And ZymoShield is something that instantly lyses cells and stops all the biology. So the RNA and DNA in those samples is preserved at room temperature for up to about a month. Again, we're trying to make our workflow as common across samples and as universal as possible, while we also try to serve the greatest breadth of samples we can. And so when we start thinking about things like leaf punches and seeds and all these other things, it's very hard for us to create a completely universally applicable workflow, right? So as a compromise short circuit on that, we also offer the ability to take purified bulk RNA. So this is in cases where we aren't likely to anytime soon to be the expert in your particular type of sample prep. You can do the extraction and we'll take care of everything else. So downstream of that, if it's cells, we do the extraction. In any case, we do the library prep, and then we do sequencing technology called 3' end counting. And I'll talk about that a little bit more in a moment. Then we do all the core bioinformatics, like deduplication and reference alignment. And we go a little bit further than that, and we provide a fairly sophisticated suite of visualization and inspection tools. Just like with the Plasmid product, we wanted to not just give you raw data, but go as far as we can to provide immediately actionable insights. This domain here, with the complexities of genome-wide expression, presented challenges that are a lot larger than what just plasmid sequencing does, where it's a more defined problem. And I've been delighted to work with a team that has really done a great job of getting you the core insights in a limited number of inspections and views that makes the workflow very simple for you, while getting a great amount of the insight. So why 3' end counting? So as opposed to more, if you're familiar with RNA-Seq, you're more likely to have run what I'm calling conventional RNA-Seq here, which is a fragmented whole transcript approach. not shown here would be another, and even less used, would be long read-based intact transcript analysis. We've chosen to go with what you see at the top because it offers a number of benefits. It is more efficient in terms of sequencing capacity because we're not distributing reads across longer inserts. And when you do, or not inserts, longer transcripts. And when you have long transcripts, you also have to do a normalization step, because if the signal is roughly proportional to the transcript length, and you have to make some assumptions about the transcript length. If you have any sort of significant alternative splicing, you have to have a hypothesis on what the actual mean transcript length is, and then try to correct to that. That introduces some error. And then for statistical reasons, just the normalization introduces some complexities as well. But with the 3' end counting approach, you get one read per transcript molecule, and you get very good linearity of quantitation. When we test this, we see transcript abundance is accurately quantifiable across 4+ orders of magnitude. And we do this using a very commonly used spike and control set called the ERCC control set and think of this case, it was spiked into HEC293 cells. So what do you get back? Well, I'm going to spend more time talking about the interactive results, but if you're just interested in grabbing your data and going, what you get is all the sort of downloadable data files you would expect. So FASTQ files containing about 20 million raw reads. We use UMIs, unique molecular identifiers. And so when we process the data, we deduplicate them, we reference align them, and you get BAM files out that contain about 10, provided you meet the RNA input requirements, which are not burdensome at all. You get about 10 million deduplicated observations of transcript molecules. Sometimes a lot more than that, in fact, but I can't guarantee it. So at 10 million, if you think about the statistics a little bit, that means for a transcript that's present at 1 count per million, on average, you're going to get about 10 observations of it, and that's enough to easily see significant changes in the expression level of that gene. And at 1 count per million, you're in the realm of protein kinases and low abundance transcription factors, and some of the more elusive but important biology that you may care to see. You also get per sample gene counts as a tab set, but basically a plain text file that you can bring into Excel or R Studio or Python or whatever you want to do with it. And you get quality control summary information from FastQC. Now, I already told you we wanted to make it as amenable to immediate user inspection as possible without requiring you to become or find a bioinformatician. Now, if you're a bioinformatician, that's fantastic. We think, and we've heard from our customers, that even if they have a lot of expertise here, these tools that I'm going to show you are really useful for getting an initial impression in minutes or seconds instead of hours or half an hour or more. And that can make the difference between deciding, oh, yeah, this is the direction I wanted to go, and tell my colleagues to proceed or not. So it can really cut out some process time. So when you get the data back, or when your results are ready, you get an email with a link in it, and you can click on that, and you immediately go to a page that'll show you something like this. And this is a dashboard of the 30 or so top differentially expressed genes across all the samples in your order, shown as a heat map. So the genes are arrayed vertically and the conditions are arrayed horizontally. And you can immediately, I think, see here how replicates are clustering very well together. And you can see that there are very obvious patterns of changes in gene expression here between these conditions. If you want to dig deeper, you can put this in a sort of tiled bar plot view, much like a qPCR assay, and immediately look at the differences at the individual gene level. So you can see just easily by eye patterns of correlation in groups, genes that might potentially be co-regulated or behaving in similar ways. And I don't know if you can make it out on the display, but you can see that there are bars, and then at the top of the bars there are also little color coded dots that correspond to that. So the bar shows the average across the replicates. The dots show the individual samples. So on a quick inspection, you can also see how much noise there is in your replicates. For example, in that very top left with CXCL9, you can see that there's very little divergence between replicates in the highly expressed 2 genes. And then when you're down way close to the noise, there's, of course, more noise. You also have tools for rapidly appraising the overall performance of the order. For example, looking at the total count of deduplicated reads that were obtained and how well they mapped. Almost always 90% or more are uniquely mapped. Some are multi-map, depending on how much your genome of interest has highly homologous variants. You also get a heat map that allows you to assess correlations at the overall gene expression pattern level across all the samples. And as you might expect, you see these medium scale blocks where replicates are all correlated to each other. And then you see, for example, towards the bottom right, you start to see areas where those blocks themselves are similar to adjacent blocks because they are undergoing similar gene expression changes. So it's a good way to get a real quick inspection on where am I seeing important changes and how well am I seeing my replicates, how tight are my replicates within that condition. And we also give you a PCA plot because everybody loves PCA plots, and your different conditions are color-coded and shown on the top 2 principal components here. And so their spatial clustering within the condition is, again, a good indicator of how tight your biological replicates were really replicating the condition. And then the spread across the dimensions tells you about the relatedness of those conditions at their endpoint. Now, of course, what you probably want to do after getting an overall sense at a really high level, I guess I should say, of your sample's behaviors, you want to look at at pairwise comparisons between individual conditions, and you want to find which specific genes are most different between them and what does that mean for the biology. So we have a console here that allows you to very rapidly assign your samples to functional categories and then create comparisons between those and launch those analyses, which take 30 seconds or so. And then those results are stored in a list you can come back to at any time. We've actually put some intelligence into this too, so that if you name your samples in a way that is at all reasonably indicative of which samples are replicates of a condition, then we'll automatically assign all these categories for you, so you don't have to do any work there at all. And we will, in fact, endeavor to figure out reasonable pairwise comparisons and run those for you before you even get the results in case they're useful. Oftentimes they are. If they're not, you can just delete them. So when that differential gene expression experiment is done, you get something like this, which is a volcano plot showing what genes are relatively upregulated and downregulated between your 2 conditions. Those dashed line axes are settable parameters. They correspond to the log to fold change you consider pertinent or not. If you, in some cases, the community default is to set that to plus and minus 1, which is a 2-fold change. But oftentimes, if you look at the log 10 adjusted p-value, which is essentially the false discovery rate for this observation, you see that there are many changes that are statistically significant but are much more subtle than a 2-fold change. And depending on the nature of your biology, you may choose to drill down further into those. So both of these axes are usable settable parameters. When you hover over any of the dots, you get information about the gene and about the statistics and the underlying quantitative information. If you click on any of them, you start populating a list. Yeah, the animation's doing it right now. You can see it populating a list on the right. And that list is what is used to populate the heat map that I showed you initially and those tile-based bar charts and everything else. Something we've added just very recently that I'm very fond of is the ability to then, and I'll come back to it more in a minute, but to save those as a defined set so that you can quickly reuse that analysis later. Currently for mouse and human, so perhaps not so immediately addressable to people in this room, we also do functional enrichment for well-defined pathways, and you can hover over those and see how those pathways are represented in the volcano plot data. You can click on them to add all the genes in the pathway to the visualization set that I mentioned earlier. And going forward, I can't promise this is a feature, but we're looking hard at what it would take to extend this approach to a lot of other species by using orthologs of pathways in central reference model organisms and then looking for orthologs in adjacent species. So I mentioned before, but once you have any genes over here, you can save that as a gene set and name it, and then that's going to be persistent across orders and sessions. So maybe you're doing a series of, maybe you're using this as a qPCR replacement and you're doing a series of experiments. You can define your control if you want to. If you want to take a housekeeping gene-based sort of baseline approach and your targeted experimental genes, you can define sets for those, do another order, and then you don't have to redefine them. You can just click on that set again and immediately get to a plot that tells you your quantitative information. So speaking more about qPCR, we were curious about how well the product would recapitulate qPCR results, and I want to take just a moment to show you some results in a model system where we were, I mentioned it before, treating HEC293 cells with beta or gamma interferon, and then looking at gene response out over 6 and 24 hours. And we just looked in the literature for some top genes that we expected to change there. And we did RT qPCR on these, as well as looking in the RNA-Seq data. And the bottom line is that the data is generally very, very well correlated. Although you can see there are a couple cases where there's some divergence. So we went back and looked at, it's a minority of cases, but there's some there. And we went back and looked at those in more detail, and to our great relief, it turned out those are all cases where the qPCR was underperforming and where the signal in the experimental conditions was not substantially different from the no template control, giving us a lot of confidence in the utility as a qPCR analog. We've also seen that being something that resonates well with customers. Molly Gasparini here was one of our early beta testers, great collaborator with us, and she's tremendous fun to work with. And she was super excited about this. She said, "This is so cool. Would have taken a lot more time to make cDNA, run qPCR." They were able to get results in just a few days instead of going through this hassle. And they were also able to. They were interested in the effect on genes like FOS and EGR1, which you can see in the volcano plot are very highly influenced by her treatment conditions. They were treating with different drugs and looking to see how these signaling pathways and transcription factors are responding. So they were able to recapitulate their qPCR-based targeted approach while also getting the much broader context and saving themselves a lot of work. Here's another example that I really like because I think it does a great job of highlighting a concept that Jim mentioned at the very beginning, which is I'll put it as the opportunity cost of getting it wrong, right? Not only when you don't have fast loops with broad insight, you can waste a lot of time. But the worst case is you make a fundamentally wrong decision, and not only do you waste time, but you perhaps risk your reputation, or you have to, you come up with results that you need to back out later. So Marie Davis here was at the Institute for Stem Cell Science and Regenerative Medicine at University of Washington, was looking at a Parkinson's disease phenotype in patient-derived lines. And in this case, the disease is caused by a mutation in a gene called GBA, a single gene thought to be causative of the phenotype. And they were attempting to test whether they could revert these diseased cells to a wild type phenotype using CRISPR to edit that GBA mutation and back it out. They were heterozygous for the mutation, and they wanted to back it out. And they had two revertant lines that they thought from other assays, from phenotypic assays, thought were essentially equivalent. And they were very much looking forward to us confirming that both of those successfully backed out the disease phenotype. And you can see maybe from the correlation map just immediately on the bottom right that wasn't the case. Revertant 2 is very different from the patient line of origin, but revertant 1 is only modestly different. So these had very different outcomes. And if they had blindly chosen to go forward with only one of these in their research, they might have had very different levels of success depending on which they chose. Instead, they spent about 3 days to disambiguate it. We're also seeing, to my great satisfaction, a lot of companies, and maybe not to, you're going to hear about a case in a few minutes for fantastic work, in my opinion, from Corey at Matza. But another collaborator of ours in the early access period, Patrick Kellish, is at a company called Kincell that's doing cell therapy work. And they have now broadly adopted RNA-Seq into all their workflows, so from the discovery process to like later production or later in their process. I'm not sure that they're actually bringing where they are in terms of bringing their cells to market, but as they've scaled up, they've incorporated RNA-Seq across their different levels. So lastly, I want to just highlight a couple of upcoming features and improvements to the service that will be coming out in this quarter. The soonest will probably be in about a month, and then the rest all before, say, into March. One thing we're really excited about is offering cross-order comparisons with batch effect corrections. So this will enable you to compare a new order to legacy orders and build upon the insight that you got from a previous order. So instead of it being a one-time transactional sort of relationship, it's more like you're building a repository of knowledge and data that you can draw upon so that your further experiments become more valuable. We're also about, when I said something within a month, this is the one within about a month. We're about to start offering ambient shipping for purified RNA. By allowing you to put it in that same Zymo Shield product that you can send cells or protoplasts in today. So right now for purified RNA, you got to ship it on dry ice. It's heavy, it's a pain. You got to bet on how much dry ice you need. It's just a general hassle. And you can't use our Dropbox network for purified RNA and dry ice. We're going to eliminate that headache. And so anybody who wants to start with purified RNA is going to have the same ordering and operational convenience that you would with our Plasmid service or with cells for RNA-Seq. And then we've seen tremendous demand for this service. And we've had to accelerate our instrument purchasing schedule somewhat dramatically. And we're in the midst of a 4x increase in our total NovaSeq X Plus fleet size, and including adding local sequencing directly in Europe. So the EMEA area, Europe and Middle East, and Asia will get the same sort of turnaround times that people are getting in North America now at as little as 3 days. So with that, I will conclude and hand it off to Corey. I'm just going to tell. I've already hyped Corey's presentation. I hope I'm not embarrassing you, but I just love this presentation so much.